I want to share basically the passion for multiomics and a couple of examples, not to the detail because some of our clients at Vugene, we still needed to write some paperwork to show you exact, metabolites or proteins, but just showing the principle of how multiomics actually is helping to uncover modes of action and mechanisms behind the human disease, and how does it help the interpretation. Even though today I think we're gonna prove it over and over. There are many different talks, brilliantly organized by biocrates. So thank you for having me. So basically, in every question, there is you with your research questions, and quite frequently they are right. What's the mode of action of a disease? Like where we can compare disease versus controls, healthy versus not healthy in that particular disease. Quite frequently, if there is a disease and if there is a treatment, we could see how treatment affects, like doesn't affect it targetly exactly what we want, or what are side effects of that, right? Like maybe there are no symptoms, but we could see molecularly how it differs and so on. And to answer these questions, people, I think, historically had a very linear approach towards multiple omics. Usually every lab has their own identity among one of the omics. Today, maybe quite a few are the identity of metabolomics. And, we sometimes say that, this lab is very good in epigenetics. This is metagenomics lab and so on. And in the best case scenario, they approach multiomics from a very linear. Let's say we have some of our clients' projects where, they're very good at RNA-seq. And I think RNA-seq is kind of everyone's most favorite, probably at some stage in their life, omic. But then you really find the limit. And then people wander into trying maybe genetics or epigenetics because it's sequence-based. People are sometimes scared to go into proteomics or metabolomics, or if you're a metabolomics lab, maybe you'll do proteomics, but epigenetics are too far away. Maybe sequentially at some point you'll get there. But this approach, linear approach towards multiomics, it's actually harder than needed because it makes biology interpretation really hard, and data integration is also hard because we have different time points, different samples, different patients, and so on. And actually, if you would, come to bioinformaticians or people who do the analysis and you discuss it, if you want to do proper multiomics, it really helps to have, let's say, paired samples. And it really helps to pinpoint, if that's actually in the translation where things happen, is it during transcription? Is it something that enzymes activity and maybe some amino acid changes, which changes how many metabolites we have? And the problem is that when we think about various omics and we think about molecular biological dogma, we think, okay, if there is a transcript, I'm just taking that as it's very commonly used, transcriptomics, RNA-seq, we think, okay, the gene was hypomethylated, so then it was transcribed from the genome. We have some mutations, maybe it's fine, we could see it from the transcriptomics. Or we think, okay, if transcript exists in a high abundance, that means there's a lot of protein, and we use it as proxy. And then we think if that enzyme or protein is there, we should see metabolomics. But we kind of suggest, and we see what's the most useful, is if you have your research question, discuss it and try to do basically as many omics as reasonable at the same time point, because then you can get precise answer, like where the changes happen, what's actually going on, because biology is messy and we already have so many different added kind of uncertainties and unknowns, right? So it really helps to do at least multiple, not all omics at the same time. And I think this sometimes does work, and then it leads to that linear progression. But why these extrapolations actually do not work? We cannot extrapolate from transcript levels to proteomics. It's not very accurate, again, to go just to see what kind of protein you have and into metabolomics and vice versa. And I think before I joined Vugene, coming from molecular biology background and bioinformatics into multiomics, I think a lot of people are not aware that molecular biology dogma, DNA, RNA, protein, and everything in between is actually not fully correct. So I'm just trying, I just want to show you today some examples why these assumptions actually do not work, and assumptions that we have within molecular biology dogma are problematic. So this is actually quite very well known that transcriptomics and proteomics levels of mRNA do not correlate with protein abundance. Of course, there might be two parts. One is very big, one biological, some are technical. And this is one of our project's data where we had a very variable sample. So even samples were very, very diverse and heterogenic, which is very common. But then they did RNA-seq and they did proteomics. It's quite a numerous proteins that they identify, but just in the numbers that you identify of what kind of genes are transcribed and translated, the RNA-seq, you get many more genes represented. The proteomics, you have twice as little proteins. Depends on proteomics. Sometimes you get even less. And if you are in exploratory stage, you want it as many as possible. So just at the very, very first step, we already have a mismatch where we see many different RNAs, and not all proteins. Maybe we see that, of course, there might be some alternative splicing and so on, but that's actually not what's there. So here we can compare what's in common in the multiomics space. But here I just wanted to draw attention that, if you do proteomics, you probably won't catch every possible protein in it. If you do RNA-seq, you're going to capture way much more, but still not everything will be captured. You might miss like 400 proteins, and that might be quite essential. So again, just it's how important it is to do, multiple omics. The correlation, one line is case, the other one control, but so it's not that much of a matter, but basically within the different samples, correlation between transcript expression and protein abundance is basically a cloud. It's not really a correlation. And then you could do, it's scaled and so on, but then if you look at log-fold change, again, it's basically a cloud. Again, it really depends on your research question and on your data set, but we can clearly see that that's not fully linear relationship. Some people might suggest, because I come from the background where I get to analyze a lot of ribosome profiling, that maybe ribosome profiling is a way to go, but answer is no. So ribosome profiling is basically do RNA-seq, you do ribosome-seq, where you see where ribosomes sit. It's really great method if you wanted to identify unknown proteins, overlapping small novel proteins. It is much closer to translation because you can infer more about translation, but you can't count the proteins right after that. So coming back to multiomics, that ribosome profiling doesn't solve that gap and issue. And here I tried to find the best possible correlation in all the publications. So this is as probably good as it can be. It is in a plant study of Arabidopsis thaliana, I think, in one strain. So 0.6 is as good as it can be. But again, it just you can't really infer if there's a lot of RNA that there is a lot of that protein. There are many things that might change that. And even if you look, and we did the same analysis for our studies where we look, which differentially expressed RNAs and which differentially abundant translated proteins there are, and if they are statistically significant. So in that case, in this case, you could see that there are proteins which have significance in difference in both RNA-seq and proteomics, but there is loads of mRNAs which are differentially expressed, and there are other proteins that are differentially expressed, but the RNAs are not. So basically, again, this is just showing how much we need to do multiomics if we want to understand what's actually happening. Of course, it all comes from, as I mentioned, biological differences. There might be, splicing. We can check that computationally. There might be post translation modifications. Sorry for misspelling the letters. There might be some proteolytic processing, RNA degradation, ribosome binding sites, low abundance, some RNA structures, which, infer with the translation, as well as, there might be experimental procedures. And in some of the projects we saw where, you develop like a cell line and do proteomics on that while you do RNA-seq on blood. So it's then very hard to compare. We could still do that, but we can try to work around this. And for biological differences, we can keep digging deeper into, but even though it is problematic if we wish to make them correlate, but actually, if you do them both, you just get more information when combined. You have a broader picture. Is this change affecting protein, or is this change affecting translation of that protein's transcript? So here it's from the same study where we also did transcriptomics and proteomics. So in transcriptomics, on the left, we do these p-value histograms where you can clearly see that there is some kind of effect, but basically in this case, heterogeneity was too big to make it statistically significant. Within group variation was too big, so we didn't get any statistically significant results. While in proteomics, we got a couple. I on purpose made them quite small, so it's unpublished, so please, yeah. I don't think that you can get too much from this. But you can see that in proteomics, we don't see too much on effect. While on RNA-seq we do, but we still need statistical power for RNA-seq. But when we did metabolomics, we could very clearly see I didn't label them again on purpose. We very clearly saw what kind of metabolites are changing. Even though there were way less metabolites, effect could be seen here. And this is actually what helped us to guide and go towards back the target genes and target pathways to build the story. And then another thing that you could do computationally very nicely, I'm calling this computationally targeted metabolomics, where we selected the specific pathway that we identified that was quite strongly affected, and we just did differential expression linear modeling, adjusting for age and sex and so on, to identify the main metabolites. And this was after treatment where we inhibit one enzyme. So it is very clear that actually when you affect the enzyme, that metabolomics is the right omics. But how does it affect everything else? You need to do other omics. And even though these people, they did transcriptomics RNA-seq as a first thing, and then they were quite surprised by this, enzymes transcript is not changed when enzyme is inhibited. But when you look at the data, you see, yeah, why it should change. So again, just very inviting you to really think, to do probably more than one omic if you want to know where the treatment or where the disease is going on. So basically here, metabolomics helped us to have a very clear start for the next steps of the analysis. Meanwhile, other omics, even though they didn't have some statistical significance, it really helped us to stratify the patients, where we can go into SNPs, or we can go into heap maps of transcripts and see that, some are mixed up samples or some need to be restructured. And we help the patient's diagnosis in these situations. The other part, which was even, I think it's less acknowledged widely, is epigenetics and transcriptomics. People do have assumption, because broadly it is true that, if something is methylated, it's kind of silenced, and, then promoter is silenced, the gene is not translated. But there is such thing, so I'm just adding this, for you to read if you want to, that there is paradoxical gene activation, that basically actually when it's methylated, then it is expressed. And here I'm just sharing the review that you can go into the details that, there are many weird biological ways of actually methylation meaning expression. And again, if you see into the data, epigenetics and transcriptomics, it's not fully correlated for sure. But with that, I'm moving to another project where we did quite a lot of different omics. And this I also wanted to draw.
Ingrida, you have 3 more minutes to talk.
Yeah. So basically that in many real life situations, we are limited by samples. We don't have, massive biobanks and so on. So, don't limit yourself by omics. And this study did by Stuart Graham, they did many different omics. And then we do the comparisons where we have epimetabolomics, epitranscriptomics, metabotranscriptomics, and so on. And one thing from multiomics integration, if you would look at this metabolite, it seems important. This gene, it does not. But when you plot them against each other, we could see that in cases and in controls, it's a different correlation. So that's one thing that you could look at. So basically, you might not have association in your controls, but you gain association in your cases. It might be other way around, or it might be that just there is correlation in both cases, but it's the opposite. And we took basically all of these associations in the middle of the circa plot. When we compared epigenetics and transcriptomics, we could see that majority of associations were gained associations in the case. And then you could identify in the circa plot small correlations that add on into important insights. And here we have epigenetics, metabolomics, transcriptomics from that dementia project of vascular cognitive impairment. Again, we have some long non-coding RNAs. We have metabolites, which correlate with many different things. And just because of doing these kind of analysis, we identified some GTPase specific pathway, and then we can dig deeper into it, add on metabolite class, and see that, sometimes metabolites in the same project, I don't share the name because this is part not published. All of this is one metabolite correlating with loads and loads on loads of differentially methylated regions and transcripts. So the VCI, we saw that there is a complex biology. We uncovered that, and this really helps to capture the subtle wide-scale biological differences. And they're now looking into maybe doing metagenomics on top of that. So this work has been done at Vugene. Special shout out to Milda in the middle, who did the analysis of the VCI part, and of course, Juozas and Gražina, who are leaders of our company. And if you are interested in how Vugene work, we work with human intelligence, not just AI. So our clients give us the data and we provide you discussions and reports. Everything from start of every omic. We do overrepresentation analysis, multiomic correlations, and so on. I didn't show too much because you can go and see all the side hustles and analysis that we do in our app.vugene.com. This is like not a commercial thing, but it's basically, if you want the details of how we do metabolomics or how we do any other omics, we have demo reports where you could see that, we go through quality control very, very thoroughly. We do, normalization, then we do another quality control. So you could see for yourself, what else you could do with your data, or maybe you can come and we can discuss. And the Vugene, we try, to help to get clearer insights and to have better science and just empower metabolomics. Thank you.